Hey guys. So originally I was just going to do the electron transport chain, this guy down here. But if we get that backtracking, we could go ahead and start from scratch with glycolysis and the Krebs cycle, and then kind of end everything, wrap everything up with what happens in the electron transport chain. So it kind of set the stage for where we have, what we have and where. We've got the cytoplasm out here. Okay, let me see cytoplasm there. Got the mitochondrial matrix, the mitochondria, Krebs cycle, and then the whole electron transport chain here. Inner membrane of the mitochondria, outer membrane, and then the inner membrane space. Lord. Forgive me, this may be kind of sloppy. I'm still learning how to use this program the best way. But we're going to learn together. So the whole thing starts again with glycolysis. So what's going to happen is glucose comes into the cell. Remember, glucose is really, really big, so glucose needs to be broken down a little bit to start this whole thing, off. Which, if we look at glycolysis, remember, glyco means glucose, lysis, splitting. So this whole step is essentially the splitting of glucose into these smaller molecules. Remember also, we're not so much worried with the intricacies of each little bitty step. We're worried about net gains and kind of how all of this contributes to the whole. So, worrying about our net gains. Remember, we've got two ATP molecules. These are going to be our big energy guys, so they're going to be used immediately for energy purposes. We also have, and this is kind of more what we're concerned with, are two NADH molecules. These guys are going to be the ones that go to the electron transport chain because these are our electron carriers. So they're going to carry these electrons over here to the electron transport chain, which we'll talk about in a second. So we have our two pyruvates. If we have oxygen, and we're going to go ahead and assume that we do, these two pyruvates are going to come in here to the mitochondrial matrix, and they're going to enter the Krebs cycle. If there's not oxygen, remember they're going to go over to fermentation. They're going to be turned into lactic acid or ethanol, depending on what organism is carrying out fermentation. Well, we're going to assume that we have O2. So when O2 is present, pyruvates coming here to Krebs cycle. There's a lot of those little intermediates in Krebs cycle that we're not going to worry about because, again, big picture. So for the Krebs cycle, we're worrying about, again, net gains. For the Krebs cycle, we got four things to get made. First, we've got 6 CO2 molecules. CO2 is going to be exhaled. It's a byproduct, essentially a waste product. If too much of it builds up, it can cause some pretty significant problems. So it needs to be excreted from the body. We're also going to have a little bit of ATP get made, just 2. Again, they're going to be used for energy purposes. Now, what's going to get used in a second are these 8 NADH molecules that we get, and also the 2 FADH2 molecules. Both of these guys are going to be coming down here to the electron transport chain because there are more of these electron carriers. And those electrons are what ultimately drive the whole electron transport chain forward. You all have to forgive me. I've kind of developed a cold today and I've been coughing a lot, so sorry. Now, these 2 NADH molecules, these 8 NADH molecules, these 2 FADH2 molecules, all of them are going to be coming way down here to the electron transport chain. Which, again, this is kind of the big show of this whole process of cellular respiration. This is the big culminating step. This is what glycolysis and the Krebs cycle are happening for, basically. Because those NADH molecules, they're going to come in here. They're going to drop off electrons. And they're going to be converted to NAD+. Same thing is going to be true of FADH2. It's going to come in, drop off some electrons, then it will be converted to FAD+. These guys, because they are electron carriers, that's the only thing they really do. So they're going to go back to glycolysis or the Krebs cycle, and they're going to pick up more electrons and then bring them right back. That's their only purpose. So, electron transport chain. Oh wow, LSU just fired Les Miles. That's going to matter to about half of you. So in this electron transport chain, because we have these electrons here, and these electrons are really high energy, they're going to start kind of slowly moving through this chain. Not so slowly. This process actually happens, lightning fast. But for our purposes, we're going to go through it slow. So the electrons will kind of start traveling through here. Now, as these electrons travel, just like sprinters, just like runners, they're going to be giving off electrons as they go. Sorry, they're going to be giving off energy as they go. Now, as they give off this energy, that energy is going to be used for active transport. All of these guys here, this big protein, this big protein, this big protein, these are protein channels. Now, they're not these passive protein channels like we talked about with facilitated diffusion. These guys are going to work in active transport. Now, what that means is whenever they have this energy, they're going to be using that energy to kind of act as a pump for these hydrogen ions. So what's going to happen is whenever this energy is given off, these hydrogen ions are going to move across this membrane down here into the intermembrane space. Okay, the same thing is going to happen with this electron that FADH2 dropped off. It's going to give off a little bit of energy. This protein is going to use that energy to take this hydrogen and move it across the membrane into this intermembrane space. And so that's going to happen at each of these three big protein transporters, is they're going to use some of the energy from these electrons to pump hydrogens across the membrane into the intermembrane space. Okay, now what that's doing, and you can kind of see this happening, is these hydrogens here, all these guys here, what we have now is we have this really high concentration here of hydrogen ions and a very low concentration of hydrogen ions in the mitochondrial matrix. Now, all of that has happened in order to serve this big final culminating step. These electrons have given off energy. Hydrogens have been pumped down here into the intermembrane space. And now we're kind of ready for this last step. Now, real quick before we talk about ATP synthase, we have to talk about how these electrons are going to get picked up. Erase. There we go. So what's going to happen is, let me get back over here. So what's going to happen is we're going to have oxygen. Oxygen's going to come into the mitochondrial matrix. It's going to pick up hydrogen ions. These two guys are going to come in here. They're going to pick up those electrons. And all of these things collectively, these three things are going to form water. Now, the interesting thing about this oxygen here, like this is the purpose of breathing. The reason that we inhale oxygen, the reason that it's so important to our well-being, is so that it can come in here and pick up these electrons. Because if these electrons don't get picked up, what happens is these electrons can get, these electrons can actually get kind of backed up in this chain. And if you have these electrons start to get backed up, then what that means is, you don't have as much energy being given off, you don't have these electrons moving as quickly, and you don't have this big concentration gradient established, which is really important. So, oxygen is extremely necessary, and you don't really think of it in that context, and mostly because you've never really heard of it in that context. But obviously, it's a really important thing. So this oxygen picks up the electrons, and it forms water. So I want to move this out of the way so I can have a little bit more room for this last step we're going to talk about. Okay, so now we're ready for this big final step. Okay, let's talk about ATP synthase. Now, all of these hydrogens, just like any other molecule, they want to go to an area of lower concentration. They don't want to be kind of cramped up close to each other in this intermembrane space. They want to have their own kind of breathing room, so to say. So what's going to start to happen is these hydrogen ions, they are going to start moving across this ATP synthase into this intermembrane space. Now, what's going to happen is as these hydrogens go through ATP synthase is going to start to kind of spin. Now, as it spins, it's going to take, let's use this color. As it spins, it's going to take ADP, which is adenosine diphosphate. It's going to grab another phosphate that's kind of floating around in the cytoplasm. And both of these guys are going to essentially get jammed together by ATP synthase. And what that does is it turns this adenosine diphosphate into adenosine triphosphate, which we know better as ATP. And ATP is the reason that we're here, basically. It's the whole goal of cellular respiration is to produce as much ATP as possible, and ATP synthase is solely responsible for that. And so to kind of backtrack and to, essentially put all this in context, the reason that we have, let's use that. So the reason that we need glucose, glucose produces these NADHs, which go to the electron transport chain. Glucose also produces pyruvates, which go to the Krebs cycle, which produces more electron transporters. All of these electron transporters are bringing these electrons to the electron transport chain. They're giving off energy. They're pumping these hydrogens across. They're establishing this big concentration gradient, which ultimately causes ATP synthase to transport these hydrogen ions, which causes it to spin, which produces our ATP. Now, for each glucose molecule, let's go right here and kind of look at our totals. Per glucose, we get 36 ATP. That's in one round of cellular respiration with one glucose kind of going through start to finish. Now, we also talked about how there are so many other electron transport chains. This whole setup here, you can find that same group of proteins. You can find another electron transport chain here and here and here. Essentially, they're all over this inner membrane of the mitochondria. So it's kind of allowing this process to happen exponentially faster. And this kind of gets to that whole form fits function thing with the kind of folded nature of the intermitochondrial membrane. It allows it to do its job really well and really effectively. So that's cellular respiration. Hopefully this helps. If you have any questions, please feel free to email me. I'm going to try to do more of these because I feel like they're relatively helpful. But any feedback is obviously really appreciated. So good luck studying for the quiz. We'll see you tomorrow in class. Thanks.